like to thank you all for coming. We are really fortunate to have Steve Carpenter here to give a second seminar. Steve got his PhD at the University of Wisconsin, and then was on the faculty at the University of Notre Dame as an assistant and associate professor. And no, I didn't forget he did not do a postdoc. This was in an era where it was possible to go straight to a faculty job. He is currently has an endowed professorship at the University of Wisconsin, and is going to be talking to us about something where he is known as sort of the expert, the guy, Trophic, Cascades, and lakes.
Thanks a lot, Alan. Thank you all for coming. Good to see everybody turning out. And I want to say that I've really enjoyed my visit here at UC Davis. I've met a bunch of interesting people and learned about a lot of interesting research. And it's a really exciting place to be. And I'm happy that Alan invited me out and that the Storer endowment was able to support the visit. It's just been really terrific. So thanks very much for your hospitality. Today I'm going to talk about some research that I've been doing for more than 30 years in experimental lakes in Wisconsin. You can see a couple of the lakes and a couple of the food webs here. And I'm going to try to give an overview of that research and point in a few directions where I think we need to go in the future with research on trophic cascades. So how are we doing? Can you all see that? I bet if we could turn off the lights in front, this would work better. And that's not because I need a nap. That's so. That was the opposite. Yeah, okay. So the purpose of this slide is to make 2 points. The first is what a trophic cascade is with the Encyclopedia Britannica definition. Probably a lot of people in the room know that it's an ecological phenomenon triggered by the addition or removal of an apex predator that leads to a series of reciprocal changes through a food web. The second reason to show this slide is to show that the term was introduced in 1980 by Robert Payne in the paper that you see here, Payne's Tansley Lecture to the British Ecological Society. So it's a relatively new ecological term. 1980 was, 34 years ago. My introduction to trophic cascades came from reading Payne's paper, but also from thinking about a problem of the time, a problem of the early 1980s in limnology. And by 1980 or so, limnologists had figured out that phosphorus was the critical limiting nutrient in lakes. A very important series of experiments had been conducted by Dave Schindler that essentially proved this. The Clean Water Act amendments of the United States were passed in 1976. Similar laws were passed in most of the rich countries of the world. And limnology had actually solved a huge environmental problem by that time. But there was also a problem that phosphorus didn't seem to explain everything. So what I've plotted here is the log of phosphorus load, that's the annual rate of phosphorus input to the lake, against the aerial chlorophyll. So that's the vertically integrated chlorophyll, that's plant biomass per square meter of lake surface for a large number of lakes around the world, some of which were experimentally manipulated in red and some of which were not in cyan. And there is a significant correlation there. R is about 0.4 or so, but there's a tremendous amount of scatter. There's more for a lake, for lakes of a given phosphorus load, there is more than 2 orders of magnitude of unexplained variability there. So there's something else going on. And at that time, limnologists were trying to figure out what it was. And Dave Schindler, who had led those famous experiments at ELA, said that eutrophication study had turned into a phosphorus bandwagon. And the explanations for that scatter hinged mostly on other nutrients, on nitrogen or more exotic micronutrients that might explain the scatter. But as Schindler alluded, maybe it had something to do with the way lake food webs were put together. And that got me and many other people thinking about trophic cascades. Today I'm going to talk to you about evidence for trophic cascades in lakes, which comes from three main sources, which I will summarize. It might be hard to believe that in 1980 this was incredibly controversial, and we were criticized very heavily in the ecological community for suggesting that animals might play a role in an ecosystem process like primary production, which was obviously controlled by edaphic factors, not by biotic ones. I want to talk a little bit about terrestrial inputs directly to predators, which I think is an important unresolved issue. I want to talk a little bit about alternate stable states and try to summarize where thinking about trophic cascades is today. So first, a little bit about evidence. In the early 1980s, we thought about trophic cascades as being a pretty vertical kind of process. So at the apex of the food web, you had piscivorous fish, large-bodied fish that ate other fish. They were subject to mortality largely by human predators through fishing. Those piscivorous fish ate smaller-bodied fish, planktivores, which ate zooplankton, which ate phytoplankton. There were some variable physical and chemical factors in there, and that was pretty much how it worked. One point I'll make through the talk is how this diagram became elaborated over time. The first experiment we did was a reciprocal transplant experiment done in 1984 to 1986. So we had a reference lake, Paul Lake here. Paul Lake has served as the reference lake for more than 30 years of these experiments, and you can remember that because Paul is small and rhymes with control, sort of. From Peter Lake, this lake here, we took 58 kilograms of bass, a top predator, and we moved them over the hill to Tuesday Lake, the lake at the top of the slide, and we moved about the same weight, about the same mass of minnows, small planktivorous fish, from Tuesday Lake into Peter Lake, and we were careful to keep the mass about the same because a lot of the phosphorus in lakes has fins on it. And phosphorus is a critical limiting nutrient. If you're moving fish, you're moving phosphorus. So those 56 kilograms of minnows, which is 49,601 minnows, by the way, were put in here, and we switched the food webs, and we, in fact, switched the zooplankton and switched the chlorophyll. We switched the primary production. It basically panned out. We did a second set of experiments from 1991 to 1997, and in those experiments we wanted to look at the interactions of nutrients with the food web. So we took 4 lakes with different food webs, Paul again as the reference system. East and West Long Lakes were piscivore dominated, and Peter Lake was planktivore dominated at the time of those experiments, and we enriched 3 of the lakes across an enrichment gradient. So we actually changed the enrichment level from year to year in a random sequence to break up autocorrelation and look at the food web contrasts and the nutrient effects at the same time. And that experiment was summarized in ecological monographs in 2001. So the bottom line of the experiments, I think, is most easily seen in these 2 graphs. On the left-hand side, you see phosphorus input rate, which we controlled experimentally, so it's known. And that's plotted against aerial chlorophyll again, so chlorophyll per unit area. And there's a positive correlation, is exactly as we would expect from the ELA experiments. There's also a lot of scatter. If you take residuals from a linear fit to the left-hand graph and you plot the residuals against mean crustacean length, and I'll explain that momentarily, you get this plot here. We picked mean crustacean length because one of the lessons of these experiments is that body size of grazers is a terrific indicator of food web structure. It's very difficult to measure fish biomass accurately. The coefficient of variation for fish biomass is usually 30-35%, and that's even if you do a lot of work to nail down the estimate. You can measure mean crustacean length very precisely. Fish are extremely size selective in eating zooplankton, so if the zooplankton are big, they are not being preyed on by fish. And if they're small, they're facing heavy fish predation. So this is an index of food web structure, and there is an inverse relationship exactly as you would expect from the trophic cascade idea in those experiments. Here I've shown you plant biomass per square meter. In fact, we looked at a wide range of ecosystem variables. So these variables on the left column, phytoplankton biomass production, total ecosystem respiration, bacterial production, carbon dioxide exchange with the atmosphere, and nitrogen phosphorus stoichiometry. These are all things that are normally, certainly in the 1980s, and were considered the purview of ecosystem ecology. Community ecologists weren't supposed to work on these. They're controlled by edaphic factors. And in fact, we showed that they're controlled by both. That nutrients, which are the standard edaphic factor of limnology, and food web structure control these ecosystem rates. And I've listed the papers there. So we feel that this showed, these two families of experiments showed pretty strong evidence at the whole lake scale that trophic cascades were approximately as important as nutrient loading in determining the ecosystem dynamics of lakes. I said I was going to give you 3 lines of evidence, and I'm going to go a little more briefly through the other 2 lines of evidence. There are a number of long-term observations that demonstrate trophic cascades, and quite a few papers in the literature by now. I'm going to tell you one that I've been involved with. And this case involves Lake Mendota, which is one of the lakes that I discussed yesterday. And Lake Mendota is the upper lake in the Landsat image there. Madison is right below it. So my office is right there on Lake Mendota. I look at it every day. Lake Mendota exhibits enormous variability in water clarity. And limnologists measure water clarity or transparency a lot of ways. One of the easiest is the Secchi disk, which was invented by a guy named Secchi, oddly enough, who was the captain of the Pope's yacht in the Adriatic. The Pope didn't use his yacht much. And so Secchi had a lot of time on his hands to do scientific studies of water clarity in the Adriatic and Mediterranean, which he did with that disk, and the disk is still used today. And the idea is you lower it into the lake and you measure the depth at which it disappears, and it tells you the water clarity. It turns out that water clarity in this lake is regulated in part by the size of Daphnia, which is an important grazer. And there are 2 species that alternate in their dominance of the lake. Daphnia Pulicaria, the big one here, is a very effective grazer with a broad diet. Daphnia galeata mendoti is a much slimmer animal and actually is composed mostly of helmet and tail spine, which are defensive structures, defenses against invertebrate predators. And Daphnia galeata mendoti has a much narrower diet. It eats a narrower range of phytoplankton species, and it has a lower grazing rate. What I've plotted here is mean log Secchi disk transparency versus annual phosphorus load to the lake in kilograms. There's an inverse relationship, as you'd expect. Lots of phosphorus means less transparency. And there's a pattern to the scatter. Daphnia pulicaria, the big one, is the solid circles, and Daphnia galeata mendoti, the little one, is the open circles. And if you plot these against time, the pattern becomes even more clear. So prior to 1987, it's mostly Daphnia galeata mendoti. After 1987, it's mostly Daphnia pulicaria. In 1987 and 1988, two important things happened. One is there was a summer kill of an important planktivore, Cisco, Coregonus artidae, which disappeared from the lake and freed the Daphnia from its predation. The second is that the Wisconsin Department of Natural Resources, working with the Center for Limnology, put 100 million predators, 100 million walleye predators, 20% of the state's hatchery capacity, into Lake Mendota in an effort to improve water clarity, and it did. And it did it largely through the food web, not through phosphorus management, but through changing the food web. Yesterday I talked about how difficult phosphorus management has been for Lake Mendota, and to date, the single most successful intervention to improve water clarity of Lake Mendota has been the Trophic Cascade food web manipulation, not phosphorus control. The third line of evidence that I want to point out is cross-ecosystem comparison. And we worked with colleagues in Ontario. Here's Wisconsin. These are the Great Lakes. This is pretty much Ontario up there. And we were able to put together data from a fairly large number of lakes that had been studied at least 2 years, most of them considerably more than 2 years, where we had data on phosphorus concentrations, food web structure, and chlorophyll. So again, similar to what I showed you for the experiments, I've plotted the log. Here I used total phosphorus concentration at spring mixes. We did not have loading for all of these lakes. Loading is really expensive to measure because you've got to do hydrology, which is intrinsically expensive. Spring mixes chlorophyll, or spring mixes total phosphorus, I'm sorry, is a pretty good index of loading, and pretty good was all we had for this study. There is a positive correlation between plant biomass and phosphorus, and there is an inverse relationship between the residual and the size of the grazers, just as in the experiments. So again, evidence for a trophic cascade effect in the chlorophyll of these lakes. So as a result of all of these studies, it seems like the response of primary producers to nutrient input rate in a stylized diagram looks like this. So if there is a 4 trophic level system with a top piscivore like largemouth bass, you get a muted response to nutrient enrichment. If you have a 3 trophic level system with no piscivore and planktivore dominance, you get a steeper response of plant biomass to nutrient enrichment in lakes. Okay, so to summarize to this point, I've showed you that both nutrient enrichment and food web structure control ecosystem processes in lakes. I've primarily used plant biomass as an indicator, but I mentioned that a number of other ecosystem responses also are driven by those same two drivers, nutrients and food web structure. Now I want to talk about a couple of complications, and the first one I want to talk about is terrestrial inputs directly to predators. Quite a while ago, we noticed that terrestrial inputs directly to predators were pretty important. So here I'm comparing the phosphorus inputs directly to several different compartments of the lake ecosystem from different sources. And the point I want to make is that these are inputs by food, by terrestrial food, directly into planktivores and directly into piscivores. So those are big inputs from eating terrestrial prey to these top predator levels. And of course there's some particulate phosphorus that's eaten by lower trophic levels. But we were very surprised at the role of terrestrial prey in the phosphorus budget. So the terrestrial prey consists, some of the examples are listed here, small mammals, snakes, amphibians, baby birds are a big prey item in spring. And these large-bodied predators just sort of sit near the edge of the lake and eat unfortunate animals that come by, and that's a lot of phosphorus and a lot of biomass. We also looked at an index of food web stability. This is from a paper back in the 1990s. And this slide, the numbers here are an index of the sensitivity of stability to different phosphorus sources. And dissolved and particulate phosphorus sources have a relatively low impact on stability of the food web. Fish prey, terrestrial prey to fishes, have a huge impact on stability of the food web according to this index. So this has, I've been intrigued by this result ever since we got it, and have in idle moments been thinking about how these terrestrial food sources affect stability and cascade strength. Daniel Schindler, son of the famous Dave Schindler, was a grad student on the project, and he used the data in a separate paper to look at cascade strength as a function of terrestrial inputs to the predators. And Daniel's argument was that cascades got stronger according to his index as you increase the terrestrial inputs. So another piece of evidence that those terrestrial inputs play an important role. Now, in the 1990s, when Daniel was doing his work and when that 1992 paper was presented, we couldn't tell littoral zone inputs from terrestrial inputs. And the reason is we didn't have a good tracer. There was no tracer to determine whether organic matter came from the littoral zone or from the land. But by the late 1990s and the aughts, there were two really good tracers. Well, there were two tracers. One of them was really good. There was carbon-13, which is very widely used, and there was a really great tracer, which is deuterium, which is very strongly different in terrestrial versus aquatic sources. And those allowed us to ask where the organic matter subsidy comes from. And of course, it could come from dissolved organic carbon, particulate organic carbon, or from various terrestrial prey. And we found that actually there's a tremendous range in the terrestrial contribution to the consumers of lakes. So here is one example from Grace Wilkinson's work. This is based largely on deuterium, and Grace worked on Chaobarus, the phantom midge, which is pictured at the top of the slide, and these are data from 40 different lakes where she sampled Chaobarus and the end members to figure out the terrestrial contribution, and you can see they go from about 5% to well over 80%. So quite a range of contribution. But sometimes the terrestrial contribution is quite high. This slide shows the proportion of organic matter from terrestrial sources to either carbon pools or consumers of 3 different lakes. This is based on carbon-13 additions to whole lakes. So in these experiments, you add carbon-13 in inorganic form. It labels up the phytoplankton and the periphyton. Then you get a very strong contrast in carbon-13 signal between the aquatic primary production and the terrestrial primary production, and you can infer the contributions. And my point here is that allochthony, the terrestrial contribution, is substantial for all of these consumers in these experimental lakes. So here's where we are with thinking about this. We know that the subsidies of organic matter are not literal. They are terrestrial. And we know that they vary widely from 5% or so to well over 80%, almost 90% in some lakes. So there's tremendous variability. We have some fairly simple results from linear models. Are you okay back there? That show that allochthonous support to the top predators stabilizes the food web, and that's principally through eating terrestrial prey items. So that's, if you're a bass, you hang around a log. When a chipmunk comes by, you jump up on the log, you knock the chipmunk in the water, you eat it, and that's a huge prey item, and then you go back and you just sit under the log for 3 or 4 days until the next chipmunk comes along. And that's how you make a living if you're a big piscivore in a Wisconsin lake. And then we also know from the Schindler work that allochthonous support to fishes make cascades stronger. And there's a recent, more sophisticated model by LaRue and Leroux in 2008 that essentially says the same thing. So I think one of the big interesting mysteries here is what's going on with stability and cascade strength across this huge gradient of allochthony in lakes between 5% and more than 80%. And I don't know the answer, but I'd love to work on it. And it's on my mind. Another complication that I want to talk about for a few minutes is alternate states and stability of alternate states of the food web of lakes. In the lakes that I work on, and I suspect in a lot of other pelagic ecosystems, trophic triangles set up alternate states of lake food webs. A trophic triangle occurs when you have some forage fish that is able to eat the juveniles of a piscivorous fish, but the forage fish is itself consumed by the adult piscivores. And the adult piscivores are subject to harvest. When harvest drives the adult piscivores to a very low density, the forage fish can escape from predation and can cut off recruitment of the piscivores by eating all of the juveniles. On the other hand, where harvest of piscivores is modest or low, you can have high abundance of adult piscivores. They control the forage fishes, and you have the piscivore-dominated state. And so you can get a flip with trophic cascades from one side to the other through harvesting this system. And there's a nice explanation of this in a paper by Carl Walters and Jim Kitchell, and I've talked about it. There's a paper by David Segal that develops some of the theoretical aspects of it in Allen's journal, Theoretical Ecology, if you want to read more about it. And I suspect these are pretty widespread in pelagic systems, certainly in lakes. A few years ago, we and Martin Scheffer and a number of other groups, including Allen's, got interested in the possibility of statistics that could announce shifts between alternate states. And so when you have high resilience of one of the alternate states, things like variance and autocorrelation in time tend to be relatively low. When the resilience of the state of the ecosystem begins to decline, you see higher variance and autocorrelation approaching 1 for the fold bifurcation, which is the bifurcation you get in trophic triangles. And we got interested in this for a number of phenomena in lakes, and used the trophic cascade as a sort of a test mechanism to explore these signals in whole lakes. And so the basic idea of the experiment was to move from an ecosystem that had a very small number of top predators to one that had a large number of top predators very gradually over time. So we essentially did a bass titration where we put a few bass in every once in a while over a period of 2 or 3 years to gradually shift the food web while monitoring a very large number of response variables at very high frequency to try to see whether the early warning signals showed up. So we measured the spatial pattern and abundance of planktivores daily for the stratified summer stratification of 4 years in the lakes. We measured the size, structure, and biomass of zooplankton daily for the 4 years. We measured chlorophyll and a few other indices of the phytoplankton daily for the 4 years. And we put SONS in the lake to measure a number of ecosystem variables every 5 minutes for the 4 years. So we had a lot of data. Here I'm showing you the variance for the daily measures of chlorophyll across the 4 summer stratified seasons, 2008 to 2011. The reference lake is in blue. The manipulated lake, the lake with the bass titration, is in red. The variance starts to increase early in the 2nd year and becomes quite high in the early to middle part of the 3rd year. We believe the regime shift was complete late in the 3rd year, and so the 4th year is sort of a back to baseline year in the different alternate state of the system. This slide just says the same thing. By now, it took, because of the massive data, it has taken quite a while to analyze this experiment, but by now we've been able to look at a number of variables and figure out what happened. The yeses mean we did see an early warning signal consistent with theory. The noes mean we did not. Most of them are yeses. I think just a couple of comments about things I found particularly interesting. The spatial pattern of fishes turned out to be a really good indicator, and we measured this with a string of 64 minnow traps in each of the two lakes monitored daily, and there were very strong pelagic to littoral shifts, and then there was a shift to schooling that showed up in spatial statistics for the minnows. Another thing I found really interesting is that net ecosystem production, gross primary production, and total ecosystem respiration, which are three of the favorite variables of limnologists and ecosystem scientists. I've spent my life learning how to measure these variables better, and they're completely worthless as early warning indicators of regime shifts. And the reason is, I think, they're derived from models, and those models have process error, and the data that go into the models has observation error, and by the time you propagate all those errors through the models, you've obliterated the signal. So they don't work. Another thing I found really interesting is that zooplankton body size doesn't work. Body size seems to be a very conservative variable that doesn't show a lot of short-term variance and pretty much tells you who's getting killed by predators, and doesn't vary much, at least in those experiments. Anyway, I think the overall picture here is that we did see a number of the early warning signals that we expected in that experiment. And I think in the interest of time, I'm not really going to go through this summary slide. Let me say a few things about status of trophic cascades in ecology. First of all, I think one of the lessons that I have taken from working on trophic cascades, and in particular from the many debates we had in the 80s and 90s about whether they occur or not, is that ecological understanding really rests on four strong legs. And in order to develop an idea in ecology, really understand it, and work it out through the ecological community, you need to think about experimental studies, comparative studies across a range of different ecosystem types, long-term observations, as well as theory and models. And I've showed you examples of all four of those. I showed whole ecosystem experiments, comparisons of lakes throughout the western Great Lakes region, long-term observations from Lake Mendota, and I could have picked many other long-term limnological studies and a bit about theory and models. And this takes time. It takes a long time for an idea to be filtered through those processes. Currently, I would say, at least my view of trophic cascades in lakes includes two important things that it didn't include in the 1980s. One of them is the importance of refuges for the planktivore, the juvenile piscivore, and the herbivore. Animal behavior and the ability of animals to hide has very important effects on the rate of trophic cascades. In some cases, it makes trophic cascades a lot faster, such as when, say, the planktivores just leave because they don't want to be in an ecosystem where they might be killed. In other cases, the behavior is able to stabilize consumers for quite a while against quite a bit of predation threat. So, for example, vertical migration by Daphnia can keep effective grazers in a system even when predation gets fairly high. Predation can reach a level where it wipes them out, but the refuge stabilizes things for quite a period of time. The second important lesson for me, at least, has been the critical role of phosphorus and organic matter flow from land to the lake. And I think the direct flow of prey items to fishes is a lot more important in stabilizing some lake food webs and in maintaining trophic cascades than we knew in the 1980s. And I personally think this is a pretty important topic for further research. One thing I hope to be doing in the next few years is take our 30-some years of data along with a few standard ecological models and begin to explore stability and cascade strength as a function of this terrestrial subsidy. We know a lot about the terrestrial subsidy because my colleague Jim Hodgson has gathered what we believe to be the world's largest library of bass barf. And he has gone through close to 10,000 bass diets from those 30-some years of research. So we know an awful lot about what those bass have been eating. Another interesting feature, I think, is that the trophic cascade concept has really taken off, and most of the papers are actually terrestrial, not lake papers. So here I've plotted publications per year that use the word trophic cascade from 1974 to 2013. There's Payne's paper in 1980, our first paper on lakes in 1985, and pretty much an exponential explosion since then. And there have been several papers that pointed out how widespread trophic cascades are as an ecological phenomenon. We published a paper in 1999 pointing to a few terrestrial examples and suggesting that it was a widespread phenomenon. By 2011, Jim Estes, John Terborg, and quite a few other folks, including me, published a paper with many more terrestrial examples, and that paper is accompanied by a book edited by Estes and Terborg that goes through the examples. Now, there is incredible variety in the way trophic cascades operate. It's not a one-size-fits-all model. There is it's not quite as bad as every case is unique, but there is incredible variability among cases, and I think there's room for a lot of cross-cascade comparative work in ecology now because of the rich variety of examples that are out there. With that, I'm going to close. I think we might have a few minutes for questions. Do we have a couple of minutes, Alan?
Yeah, we can go till the other room.
Oh, okay. Don.
So, the oceanic food webs work the same way, and they don't finger phosphorus. They've got the trophic triangle, they've got big, huge piscivores, middle-sized piscivores, what they call forage fish.
Uh-huh.
So, has anybody looked at the difference between these oceanic ones and your lymphatic ones?
No, and I think that would be a really interesting thing to do. An obvious contrast is the role of the terrestrial input in the lake cascades. The perimeter to area ratio for lakes is obviously much more favorable for those terrestrial inputs. I think another interesting aspect might be the medium to large scale spatial heterogeneity of marine ecosystems and the capacity of animals to move fairly considerable distances. For example, Jim Estes talks about killer whales shifting from the open sea in the Bering Sea region to a more inshore predation on marine mammals, causing cascades inshore. That's a habitat shift that I I have no knowledge of in lakes. I haven't seen an analog of that in lake ecosystems. So I think it's a great question. I would look at the role of terrestrial inputs and meso to macro scale habitat shifts of top predators. What do you think, Don?
Bird shit.
Yeah? You think
Seriously.
in the near shore?
Well, even pelagic, I mean, there's a recent paper in MES on pelagic inputs of bird food.
Uh-huh. Now where, I mean,
That's just a guess.
are those pelagic birds eating pelagic fish, or are they really carrying terrestrial?
The author's probably here.
Anybody want to own up to that?
Why doesn't he weigh in on it?
Yeah. So what's if you're an albatross? Where does your poop come from?
Well, it comes from the prey they eat, squid, fish, krill, stuff like that.
Uh-huh.
So what we were looking at was just how to recycle it in the marine environment, right? So,
Okay.
So limiting nutrients out in the pelagic ecosystem are different than limiting nutrients in the terrestrial ecosystem and the nearshore ecosystem. So we're concerned mainly with oceanic pelagic productivity.
Okay.
I think it could be interesting whenever birds or anything are pooping on your lakes.
Uh-huh. Yeah, that is an interesting thought. Certainly, ospreys and loons eat a lot of fish from.
I don't know if they poop at your lakes or on land. That's also.
Loons have some sight fidelity. Ospreys are all over the place. The osprey that was the main predator on our lakes had a nest about a half a mile away. We know because we go over under that nest to get our Floyd tags back.
Yeah. Cool. Thanks, Dan.
On the issue of, it's an interesting contrast for me as a behavioral ecologist doing community stuff that part of the trophic cascade, maybe a fair part of it, is behavioral rather than outright the bass eat the minnows. Has the big difference that, of course, if a lot of it is minnows shift and go and hide, then bass don't get to eat much. But that actually folds in that the part where they get the subsidy from land, being an important part of what keeps them up, actually ties together two of your big points there, right? That,
Yeah, that's a really interesting point. And when minnows get scared, they go inshore and they find logs and macrophytes to hide among. That draws the predators inshore, where they can find chipmunks and baby birds, as well as the occasional fish. And so maybe that behavioral response by fish is what makes the littoral zone such a hot spot for predator-prey interactions. Yeah, interesting point.
So, on the early warning signal stuff, you have a system that you understand pretty well, and you have a reference link. How would it work if you, in some sense, didn't understand the system so well and you didn't have the reference link?
Yeah, well, I think it would be a lot harder to make the correct inference from the statistics. I mean, obviously, before we did this experiment, we published a fairly detailed food web model calibrated against something like 28 lake years of data that showed what signals we should get, and we got most of them. And without that model, we could have easily been wrong. So I think that we have a long way to go before these statistics are ready for ecosystem management in a situation where we don't know what's going on. And in fact, I think that well, in my opinion, the biggest research need for these early warning signals or resilience indicators is more experimental work. And I think it should be done in experimental settings where people can build a pretty good mathematical model of what's going on and have references as well. Yeah. Yes?
So there was a fascinating paper published recently that dug up like 600 or 700 years of resting eggs of daphnia from a lake in Minnesota and showed that, kind of coincident with the onset of industrialized agriculture in the area, their phosphorus utilization changed drastically and some other things. And I'm not really sure about what the consequences of that would be for some of the stories you're telling, but maybe you could comment on the idea that maybe some of the fauna in areas that are more agriculture or more in cities are evolved to those kinds of pressures, and how that would affect the sort of
Right.
generality of findings from these experimental.
Yep, I remember that paper that was in Ecology Letters, I believe, and again, if my memory is correct, it showed very rapid changes in physiological capacities of the Daphnia and I believe there was a genetic, there had actually been selection for different physiologies of the Daphnia that was quite fast. And so that, well, I guess, fish are still going to like to eat big items, right? So fish will still operate in a size selective way, pretty much the way I've seen in my data sets. On the other hand, the predator avoidance behaviors of Daphnia might change, and the ability and perhaps the phosphorus recycling characteristics of Daphnia would change. One of the big surprises of our experiments in the 1980s was that big Daphnia retain phosphorus and recycle nitrogen, so they shift the N to P ratio toward P limitation. Jim Elser was a student with me at the time and published that in a paper in L&O in 1988 that in some ways was the beginning of ecological stoichiometry. And maybe that would be affected. I mean, obviously the northern Wisconsin experimental lakes Daphnia had never seen a cow. And the Daphnia that I showed you from Lake Mendota are experiencing a very highly polluted agricultural system. Yeah, good question. I don't have an answer other than that I've seen trophic cascades in both culturally eutrophic and fairly oligotrophic lakes.
Did you look at the phytoplankton communities and how they changed with the phosphorus load?
Yes, and maybe I should ask what specifically your question is.
I guess I was thinking, that with any addition or subtraction of phosphorus, you're going to get huge changes in the community of phytoplankton. And the Daphnia in that particular lake are adapted to eating certain food sources, and when that changes quite drastically, that changes the food sources. And I don't know what sort of an effect that they can even starve in some situations where things have been moved around too much.
Well, I can say that I don't see any evidence that Daphnia have starved in our whole lake eutrophication experiments, nor do I see any evidence that they're starving in Lake Mendota. The evidence I see there is fish regulate the Daphnia pretty strongly, and in Lake Mendota we have huge colonies, and the Daphnia just navigate around them. Burge, in his famous series of monographs about the plankton of Lake Mendota, talks about Daphnia in Lake Mendota I should preface this by saying Lake Mendota was culturally eutrophic before Burge got there, because raw human sewage was being dumped in from about 1860. Burge talks about the Daphnia manipulating 5 millimeter long segments of Anabina and jamming them down the throat of the Daphnia and piling them up in the gut. Just beautiful descriptions, verbal with hand-drawn sketches in those papers from the 1880s. So they can handle pretty large particles. Andy?
When top predators suppress
forest minnows, what actually in these systems keeps invertebrate predators, chaobrus, or anything else from actually getting abundant enough to have major impacts on the Daphnia?
First of all, Daphnia form neck teeth, which is an anti-predation morphology. So that helps. The second is, I think the second factor is that the chaobrus that occur in July and August were oviposited in early June, or in some cases in autumn the year before, and they may be, the numerical response of the chaobrus may be uncoupled due to that terrestrial life phase from the abundance of Daphnia prey. Daphnia are not very common in chaobrus guts. Patricia Serrano, when she was an undergraduate student of mine, did very painstaking analyses of chaobrus guts, and they don't eat very much Daphnia, so those anti-predator morphologies are effective. They're just packed with copepods, just like little sausages, bam, 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 stacked up in the gut.
So it seems like the traits of your primary consumers, right, the size of these food stations is really important in explaining things. And it seems like that can be explained, the shifts in that, either by the community, right, the proportion of Daphnia species, or by demography, right, less Daphnia reaching maturity, or by some evolutionary response to predation. So I'm curious if there's any way to sort of break those different components apart and see how important the evolvability of those primary consumers are to those stability of these trophic structures.
Boy, I think that's a great question. I'm not aware that anyone has done it. There is a tremendous amount known about the population genetics of Daphnia. Paul Hébert and others have done an enormous amount of research on that, but I am not aware of how selection within Daphnia pulicaria might affect these phenomena. There are half a dozen Daphnia species that are important in the lakes that I talked about, and they range in adult body size from about 1.5 millimeters to 0.4 millimeters. The smaller ones are not very heavily favored by fishes. There are even copepods that are larger than 0.4 millimeters. So fish predation has a pretty strong effect on which of those 5 or 6 species will be dominant. As to selection within the species, I don't know. That doesn't mean that nobody knows. That just means I don't know.